Chemoresistance caused by low oxygen levels in the tumor microenvironment is an ongoing challenge in cancer treatment. Fortunately, we can use polymerized hemoglobins to effectively reduce hypoxia in the tumor environment via oxygen delivery. Hi, my name is Don Belcher. In today's talk, I'll be showcasing how the oxygen affinity of polymerized hemoglobins and the tumor oxygenation status play important roles on determining the hypoxia reduction of these materials.
When considering the oxygen delivery to tumors, I like to think of three levels, here shown on the screen as the anoxic, hypoxic, and normoxic conditions. For the anoxic cells, nutrient deprivation and a lack of oxygen cause cell death. However, if we try to treat the remaining cells with chemotherapy, the hypoxic cells produce hypoxia-inducible factors which induce chemoresistance that would normally lead to cell death in normoxic cells. So, considering this, a key target of many scientists has been the oxygen delivery to tumors. The first method is normalization. This is a method popularized in the early 2000s through the use of anti-angiogenic code therapy such as Avastin, which normalizes the tortuous vascular environment of the tumor to hopefully restore oxygen delivery. However, recently there's been some doubt with these methods. US FDA has determined that this may be ineffective in the case of metastatic breast cancer. The second and by far easiest method of oxygen delivery is intensification, in this way putting the patient on a carbogen or hyperbaric based code therapy, which increases the total oxygen supply to the tissue. However, it can require long exposure times and may lead to oxidative stress. The final method, and the one I'll be talking about today, is regulation. Here, transfusing an oxygen carrier that modulates the way that oxygen is delivered to the tissue. Here, red blood cell substitutes are an excellent candidate for regulating oxygen delivery. Now, there's a wide variety of materials that can be used for red blood cell substitutes, from perfluorocarbons to encapsulated hemoglobins to even non-mammalian hemoglobin type species like erythrocurins. However, for today, I'd like to focus on polymerized hemoglobin. This is a relatively common red blood cell substitute that is cheap to produce and is relatively well characterized. In terms of polymerized hemoglobin synthesis, what we do is prepare two species, either the R state high affinity oxygen carrier or the low affinity T state. Both of these methods are prepared by first locking the hemoglobin entirely in either the R or the T quaternary state prior to polymerization with a glutaraldehyde polymerization with a sodium borohydride cyanoborohydride quench. What results are carriers with fundamentally different properties, and these properties translate to how they bind oxygen. So here we have differences in oxygen binding between humans, mice, and of course the polymerized hemoglobins. What we can see is the R state has a very high oxygen affinity, not offloading oxygen until in hypoxia, whereas the T state's low oxygen affinity gives relatively even oxygen offloading across the entire spectrum. Now, what does this mean for oxygen delivery to cancer? So, first, we'd like to consider normal red blood cells. In this situation, we expect a lot of oxygen to be delivered under the normoxic conditions and less under hypoxia. And on this plot, the black dots indicate the total oxygen release is measured by a oxygen consumption rate in a crow tissue cylinder model, and the green triangles are the red blood cell hemoglobin. Now, if we were to simulate the addition of a T-state polymerized hemoglobin, what we would notice in the model is a systematic increase of the oxygen delivery from the normoxic to the hypoxic conditions. In contrast with the R-state polymerized hemoglobin, we see an overall decrease initially at high normoxic conditions, and you get lower and lower. That spikes at around 3 millimeters mercury. So this indicates that T state may be able to selectively deliver oxygen in the tumor. As stated previously, this was done with a crow tissue cylinder model back in 2018, but we wanted to move forward and beyond that, so we looked at simulating things a bit more advanced. And for this, we utilized Tumor Code. This is a freely available tumor simulation framework where we can simulate relatively complex vascular structure in highly complex, uh, very biorealistic vascular networks connecting arteries and veins with capillary networks. Here, arterioles are red, venules are blue, and this has a large 6 millimeter cube. Once we have these cubes of vessels generated, we can then simulate tumor growth in the tissue. And on the right, you can see a tumor. This will be growing over 40 days. And on the left are some of the properties plotted over the radius from the tumor center. Things like the percent of tumor cells, the microvascular density, and the average partial pressure of oxygen. As the tumor begins to grow and expand, we see that initially a depletion of oxygen in the center due to increased respiration of the tumor cells. This is followed by a decrease in microvascular density with an increase around the edge due to internal stresses and angiogenic factors, which in the end results in a rather heterogeneous, complex tumor structure, which can be interesting for us to understand some of the spatial, temporal sort of combinations of different components. And we can actually generate this for not just one, but many different tumors. Here is a sample library of tumors, 9 different configurations, 7 different tumors, or 8 here for each different configuration. And we have a lot that we can work with. These scale bars on each plot is about 1 millimeter. So these are some large tumor structures. The green indicates tumor, red is necrotic region, and blue is for the vasculature. So with these different tumors, we can effectively simulate the oxygen transport in the system. Here, we look at various comparisons. The violin plots show the simulated tumor data, and then overlaid on top is a box plot, which is the corresponding experimental data as measured from a murine dorsal chamber window model. Here, looking at a hypoxic and a normoxic tumor. So looking at these, this is a relatively good approximation of the different tumor conditions based off the different HBOCs that can be delivered. And continuing looking at this, we can then simulate how various factors are looking in the tissue, and we can actually create different simulation states, whether based off of mouse or human physiology. Key differences being going back to our oxygen equilibrium curves, differences in how mouse and human blood offloads oxygen, and in how the oxygen in the tissue is consumed by the cells. So what we can see is differences in the predicted boundary tissue oxygen content, but more interestingly for us here is a significant decrease in the boundary hypoxic volume in the tissue after treatment with the oxygen carrier. And if we continue, we could actually look not just at the oxygen levels or the volume of the hypoxic tissue, we could also look at the oxygen extraction fraction. This is if we were to imagine the hemoglobin going into and out of the tumor, what percent on average is extracted. We could see that in general, the human hemoglobin has more of the oxygen extracted than the mouse hemoglobin, but more interestingly, in the polymerized hemoglobin, we see that much more of the T state polymerized hemoglobin has its oxygen extracted compared to the R state, especially in humans, which is expected due to the differences in the oxygen affinity. So, knowing all of this, we decided to pursue some basic experimental studies. So, looking here at a normoxic and a hypoxic melanoma in a mouse model, where we had mice with the FME hypoxic cell line and the LOX normoxic cell line. The
and the tumors were allowed to grow for 2 weeks, followed by 2 weeks of a weekly administration, 20% blood volume of either Ringer's lactate as a control, T state, or R state polymerized hemoglobin. So, first revisiting the oxygen extraction in the animal model, we noted that in the chamber window, the oxygen extraction was considerably higher. This is likely due to the two-dimensional geometry restricting the blood flow of the vessels to be uh, across the tumor structure. We found similar trends where the T state far outperforms the R state in delivering oxygen. In fact, in the normoxic tumor, the R state has practically no contribution. This translates to interesting results in the mouse model, wherein in the hypoxic tumor, the T and the R state decrease the growth, but interestingly, in the normoxic tumor, both of these species actually lead to an increase in the tumor growth. This is accompanied by corresponding increases in the regional blood flow in the tumor, and for the R state, an increase in the microvascular density. Curiously, the T state does not have any significant impacts in the microvascular density. However, there still is an increase in blood flow. There's this interesting contradiction here, likely due to some amount of changes in the angiogenesis, and we expect potentially that there may be some regulatory effects going on, and perhaps the R state being unable to deliver oxygen under normoxic conditions may be leading to increases in the angiogenesis in the tumor. But we wanted to keep looking at the R state because previous studies have shown that R state polymerized hemoglobin can be used to treat tumors to some degree. So we wanted to explore what may be going on there, if not oxygen transport. And one mechanism we decided to look at is ROS, so reactive oxygen species production. Here, this is with a series of non-small cell lung cancers, H23, H460, and A549. What we found is that cisplatin in combination with polymerized hemoglobin led to a significant decrease in tumor cell viability. And whenever we looked at actually imaging the reactive oxygen species, we found there was a significant increase in these species after the combination with polymerized hemoglobin. This is likely due to the heme in the polymerized hemoglobin, which will auto-oxidize and begin to release reactive oxygen species like hydrogen peroxide and superoxide into solution. Now, looking at how this translates into a model, we looked at a combination treatment of cisplatin and polymerized hemoglobin and found that combining the two led to significant decreases in tumor growth. However, as anticipated, cisplatin and polymerized hemoglobin don't appear to significantly affect the expression of hypoxia inducible factors, which is again another indicator that likely the source of this is not related to the oxygenation in the system. In conclusion, the T-state polymerized hemoglobin or any other low affinity oxygen carrier will likely be significantly better at oxygenating tumors. Additionally, R-state polymerized hemoglobin may have increased performance if the tissue blood saturation is less than 20%, or potentially due to ROS generation. The developed 3D models in these tumors were interesting in helping to predict the tumor oxygenation, which may help predict therapeutic response during clinical translation. And finally, the HBOC treatment on mice should be carefully considered. We observed there were significant changes between the human and the mouse physiology in our 3D models. I'd like to take a quick moment to acknowledge everyone at the Andre Palmer Research Lab for all of their help over the years with doing these experiments. I'd also like to thank our collaborators at The Ohio State University and UC San Diego for their help
with some of the experimental studies performed here. Finally, I'd like to thank our various funders, the NIH, Pelotonia, and the Ohio Supercomputing Center, without which none of this work would be possible.
Thank you for taking the time to listen to my talk today. If you have any questions, please reach out and I'll answer them as soon as I'm able. Thank you again and have a wonderful day.